I don't think there is any function. Why they are so much absent? What do you want? Should I send you out? I am telling you. As in previous classes, we had talked about the stomach, esophagus, duodenum, the small intestine, which begins from duodenum and continues up to the cecum, where it terminates into ileocecal valve. Cecum onwards is a part of large intestine. Cecum, colon, at the ascending colon, transverse colon, descending colons, rectum, and anal canal. Up to that part, that is a part of large intestine. When we see the small intestine, it is important to identify the part of small intestine with microscope as well as with naked eye. I am telling you because once you become surgeon, you must identify that the GIT when you open, when you will do the dissection of the abdomen and this part, you will find that as soon as the abdomen is open, the GIT is spread. The same thing will happen while the time of doing surgery. So you must differentiate that this is the part of the small intestine, this is the part of large intestine. So basic thing, how we identify that this is the large intestine, this is the small intestine by naked eye. When we open the or do the peritoneum, abdomen, the abdomen, what we find that the small intestine is showing the large number of permanent folds. It is not looking straight. It is showing the large number of permanent folds. I am using the word permanent folds. These permanent folds are called plica circularis. These permanent folds are called plica circularis. And the plica circularis, it is consisting of mucosa plus submucosa. Underline that. It is commonly asked in MCQ. Plica circularis is also named as Kerkering valve. Kerkering valve, Kerking, Kerkering valve or the plica circularis. What are these? These are the permanent folds which are consisting of mucosa and submucosa. All right. When we see under the microscope, I will use the word villi, large number of the villi. Singular is villus. Villi are finger-like projections. Villi are finger-like projections. What happens that mucosa of the GIT is folded. It is showing the projections, and these projections may be leaf-like, may be finger-like, or maybe the tongue-like. All these are projections are called villi. Understand? So one part of the mucous membrane is projecting outward, that is called villus, and the remaining part of the mucous membrane is going inward in the lamina propria. Understand? The part of the mucous membrane which is projecting or invaginate in the lamina propria, that is called crypts of Leverkuhn. That is called crypts of Leverkuhn. You may say, सर स्टोमक में जो म्यूकस मेम्ब्रेन इन्वेजिनेट होती है वो भी तो इन्वेजिनेट होती है लेमिना प्रोप्रिया. All right, but there we name these as gastric pits. In stomach we name those as gastric pits. In small intestine these are called crypts of Leverkuhn. All right, and these villi you can see only under microscope, not macroscopic or not with naked eye. Understand? Maybe macroscopically you can see the plica circularis. The difference between the plica circularis and villi, in plica circularis, the mucosa and submucosa both are present, while in villus only the lamina propria. Only the lamina propria, or may also some part of the smooth muscle fibers if required, nothing else. There is no submucosal part. Understand? In villi, there is lamina propria. There may also the smooth muscle fiber may extend from muscularis mucosa if the requirement is there, but no submucosa. While in plica circularis or Kerckering valve, both mucosa and submucosa are present. Before starting the part, I also want to revise by seeing your condition that you forget each and everything, or you are not giving importance to the education or study. Mucosa part is consisting of 3 layers: epithelium, lamina propria, and muscularis mucosa. These 3 are combinedly named as mucosa. Below the mucosa, I am telling you the general plan of the GIT. First is mucosa. Next to the mucosa is submucosa. Next to the mucosa is submucosa. Next to the submucosa is coming the muscularis externa. Outer the muscularis externa, there is adventitia. Adventitia is a part of loose connective tissue. As we use the word loose connective tissue may contain the blood vessels, or depending upon the part, blood vessels and lymphatic nerves. All right. When we see the structures, all, but in case of retroperitoneal organs, that means the organs which are lying behind the peritoneum, not covered by the peritoneum, those are called retroperitoneal organs. These retroperitoneal organs, kidney and all these retroperitoneal organs, what happens that over the connective tissue, there is no simple squamous epithelium, which is called the seroja. But all the structures which are not retroperitoneal, lying over the connective tissue, there is also present the simple squamous epithelium, seroja. If you can remember, very good. Not, doesn't matter. Now the point comes, jejunum and ileum. There is basic difference. The parietal cells, they start before the other cells. I want to tell you the differentiating. अगर आपको बात करनी है बाहर चल जाओ please. Don't show me your faces. If you are not interested, get out. I am not taking attendance. Don't sit here for the sake of attendance. The number of the goblet cells are increasing as we go down from duodenum to the large intestine. One thing. There is no goblet cell in the stomach. Have you seen, those who have seen the slide of the stomach, have you seen the goblet cell in the stomach? No goblet cell. But they start appearing in the duodenum. As we go down, coming from duodenum to jejunum, ileum or large intestine, there is large number of the goblet cells. Second point, differentiating point. In a small intestine, you will find villi and crypts of liver coon, both. While in large intestine, why don't you write? क्यों बेवकूफी तरह बैठे रहते हो? Then लिख लोगे तो भूल जाओगे क्या? Shut up. In large intestine, no villi, only crypts of Leverkusen. So I told you 2 points, very important points for identifying the large intestine. No villi, only the crypts of Leverkusen, and number of the goblet cells are too much, then their number is more than the columnar cell. I had already told you about the duodenum, so I will start from the jejunum. Look, I told you the plica circularis, that these are the plica circularis. These are permanent folds, and this is the villus. Sometimes I had seen that twice or thrice time the question asks, what are the contents of villus? So you give the diagram. Look, the contents of villus. In the center of the villus there is the lacteal. Lacteal is the lymphatic vessel. Lacteal is present in the center, and that is the lymphatic vessels. The function of the lacteal is absorption of the fat. Absorption of the fat. Nearby the lacteal there is present the arteriole, and on the other side there is present venule. And the rest of the part which is present or shown here in the diagram, what is that? Lamina propria. Okay, nothing else. So this is the structure of the villus. In a small intestine, large number of the villi are present. Now the next question, what is the use of villi? To increase the surface area. To increase the surface area, function of the small intestine and this absorption of the important food, and for the absorption it requires that the food should remain for longer time and for the larger area. Because of that there is present the villi. Now look, when we see the section of jejunum, in jejunum every part, jejunum, ileum, there all you will find the villi. One thing. Now what is in the villi? Look that part. Villi, each villus is consisting of columnar cells. The columnar cells are shown. Lying between the columnar cells, these are the goblet cells. Here the nuclei are not shown, but don't draw. When you draw in your record, empty goblet cell, and in the basement there draw the nucleus, flattened nuclei. Okay? Don't tell me, sir, it was not shown in the diagram. So this is the part of the villus. Now look, the invagination inside the lamina propria, that is this part. These are the crypts of Leverkusen. These are the crypts of Leverkusen. As we see the crypts of Leverkusen, in the crypts of Leverkusen there are number of the other cells are present. Besides the goblet cells and columnar cells, there are other number of the cells which are shown in other colors. The color which is shown here, pink color or the slightly reddish color, that is called Paneth cells. Paneth cells are pyramidal in shape. Paneth cells are pyramidal in shape, and they are containing the eosinophilic granules. They are secreting the enzyme lysozyme. Next to this, that is the yellowish color. These are the cell enteroendocrine cells. Enteroendocrine cells which are secreting the hormones. Enteroendocrine cells. लिख लो, कुछ दिमाग में बैठेगा, वैसे ही साफ है. क्यों नहीं लिखते हो आप लोग? Third type of cell is the stem cell. Stem cell, which is not shown here. Stem cell function is the GIT, epithelium of the GIT is vanished or removed within 5 days. So it requires the replacement. So replacement of the lost epithelium is done by the stem cells. So these are the different type of the cells which are present in the part of and crypts of liver cool. They are present in the crypts of liver cool. All right. We will see the villi and crypts of liver cool in the part of jejunum, ileum. Now the point how we differentiate also seen in the duodenum. The third point, which is not shown in the diagram, lying over the epithelium. Lying over the epithelium, you will see there is a pink color. As if you remember, also seen in the duodenum. Lying over the epithelium, there is pink color line. That pink color line is the brush border. Again, the purpose of the brush border is to increase the surface area. And this brush border is also called microvilli. Also called the microvilli, which are forming the striated border or brush border better, you remember. We use the word microvilli when we see under the electron microscope. That means they are seen very low finger-like processes. But in our slide or microscope in which we are seeing the slide, no microvilli are seen. They are seen just as a pink color line, brush border. The submucosal part. The submucosal part is showing neither the Brunner's gland nor the Pearce patches. I am talking of the jejunum. In jejunum, you will see the villi. The villi, I have never noticed the shape. They have mentioned that in the duodenum, the villi are leaf shape, but I never give importance to the shape. In the part of villus, they are tongue shape. That means slightly larger. And in the ileum, they are again finger shape. Doesn't matter. The important part that in the case of jejunum, the villi are longer, larger in number. Villi are longer in size and larger in number, and the submucosal part of the jejunum is neither showing the aggregation of the lymphoid tissue. I am using the word no aggregation of the lymphoid tissue. Second thing, nor the collection of mucus secreting glands, that is Brunner's gland, which are seen in the duodenum, and no aggregations of the lymphoid follicles, which are seen in the ileum. Those are called Pearce patches. So no Pearce patches, no Brunner's gland in the submucosal part. In submucosal part, you will see just the transverse section of the villus and all that. The muscularis externa is same, that is inner circular and outer longitudinal. And please, one thing kept in your mind, I will not use repeatedly. Whenever you describe or giving answer the question, mention that in submucosa there is the nerve plexus, which is called Meissner's nerve plexus. Don't forget to write those. In muscularis externa, between the two layers, there is another nerve plexus, that is Arbach's nerve plexus or Mantic nerve plexus. So while giving answer, mention these things definitely. Okay? Then come to the adventitia part. Same, loose connective tissue and also the part of the maybe the blood vessels. Now look the slide. That is the picture seen in the microscope. You are seeing the columnar cells. What are these? These are the villi. In each villus, columnar cells, lying between the columnar cells, the goblet cells. Okay, and this is the lamina propria, rich in the loose connective tissue, the some part of the muscle, smooth muscle fiber also seen, and these are the intestinal glands, transversely cut intestinal glands. This is the muscularis mucosa, and this is the part of lamina propria, and this is the inner circular and outer longitudinal muscle cord. And below the muscularis mucosa, this is the part of submucosa. So submucosa is empty, not empty, but having no collection of the mucus secreting glands, and the remaining part, muscle coat and everything, they are clearly, and this is the seroja. Now you understand the word seroja clearly, that is simple squamous epithelium lining the adventitia. So this is the picture of jejunum. Now look, again you are seeing the villi. Between the villi, the number of the goblet cells compared to the jejunum are more. As we go down, the number of the goblet cells increasing. And the lamina propria, and look this part. The lamina propria part and go down, also the interpret the muscularis mucosa, that is aggregated masses of the lymphatic tissue. In ileum, the aggregation of the lymphatic tissue is too much, that it is present in both lamina propria part and also the submucosal part. And you can see also here the interrupted muscularis mucosae. And this aggregated masses of lymphatic tissue is called Peyer's patches. And look the different type of the cells which I told you present in the GIT. These are the simple columnar cells. You can see these are the panic cells which are showing the eosinophilic granules, and between them these are the enteroendocrine cells which are secreting the hormones. The stem cells are not shown here, they must be present. Now look, this is the slide of colon. There is no villi. Have you seen any evagination? No. These are the invagination. That means only the crypts of Leverkusen are present. And number of the goblet cells increasing or not? Number of the goblet cells increase, and no part of the villi. Look the lamina propria. No special features. Slightly you can see the lymphatic tissue. Lymphatic tissue may be seen, but not so aggregated as seen in the Peyer's patches. Now one thing, look the muscularis externa, inner circular, outer longitudinal. Now one thing which you must mention, in the large intestine, the outer coat or longitudinal muscular fibers, they are collected in the three bands. It is not so clearly, otherwise the longitudinal muscle coat in large intestine, they are collected at the three bands, and these three bands are called tinea coli. The tinea coli can be seen with naked eye also. So we can easily differentiate large intestine from small intestine by these features. And the another for differentiating large intestine and small intestine, I will show you while you doing the dissection. But keep this thing in your mind if you can, that on the surface of the small intestine, the muscularis mucosa is extending out, villus part is remaining inside, and the superficial surface is also showing the folds. These folds are containing the fat cells. These are named as appendices epiploicae, that I will show you in the dissection hall. On the outer surface of this, there is present appendices epiploicae. Okay, at present you remember, while seeing the slide in the microscope, we will find these features. No villi, only the crypts of Leverkusen, lamina propria, muscularis mucosa, submucosa, and the muscularis externa part, that is longitudinal muscle fibers, they are collected at the three places, and these are called tinea coli. Tinea coli is special features by which one tinea coli, another novili are the special features for the large intestine, colon. Then one more slide of large intestine we will show you, that is appendix. In appendix, starting from inwards to outward, the lumen is showing again by the epithelium, columnar cells. Between the columnar cells, the goblet cells, same feature, and you can see the lumen very clearly in the slide. Outside, below the epithelium, there is a part of lamina propria. In the lamina propria, there is present the abundant amount of lymphoid tissue. You can see the very large amount of the lymphoid tissue present in the lamina propria. That is also interrupted in the part of submucosa. The same picture seen in the colon. Then if someone asks you, then how you will differentiate the slides of colon or appendix? The differentiating point, in the slide of appendix you can see the lumen. The no lumen is seen in the colon, one. Another thing, that muscularis externa of colon is collected at the three bands. That means tinea coli. Tinea coli are seen in the colon, not in appendix. These are the differentiating points by which we can differentiate the slides of colon or appendix. Okay, so when I ask, I hope you will reply. Otherwise, we cannot differentiate that the slide is of appendix or the colon. So these are the slides of GIT which we will show you. Now the another point, the accessory glands associated with the GIT. I am talking of those. देखो बाहर है क्या वो? अब वो दूसरी वाला चेंज कर दो. अच्छा वो आप आ गए. ये तो हो गया. हाँ वो दूसरी ये वाला जो खुली हुई थी क्या? पता नहीं मेरे को. Now we are talking of the glands associated with the GIT. Liver is the largest gland of the body, and this largest gland of the body, the slide of the liver is very easy to identify. It is very simple to identify, and keep one thing in your mind, as you will get the easy slide which is easily identified, the biopsy is so much tight. The function of the liver, it is receiving two types of blood supply, from the portal vein and from the hepatic artery. The blood supply returning from the hepatic artery is about 20%, while the blood supply receiving from the portal vein is about 70 to 80%. Now the first point, what is coming through the blood supply of the portal vein? That is the blood which is coming from the portal vein, receiving all the metabolites which are taken out from the digestion of the foods and this. So important metabolites which the blood from the GIT goes to the liver, and oxygenated blood is through the hepatic artery. The liver is surrounding by the capsule, connective tissue capsule, and that connective tissue capsule is called Gleason's capsule. That is called Gleason's capsule. the septas passing inwards, they divide the liver into the lobules. You can see this is the one lobule, this is another lobule, like that. Each lobule, that means when we see the interior of the liver, the capsule, from the capsule the septas are coming inwards, and these septas dividing the liver into the lobules. Each lobule is having the vein in the center, that is this one, that is called central vein. This is called central vein, and radiating from the central vein you are seeing the multiple cords. देखिए ये central vein है. Radiating from the central vein there are multiple cords, and each cord is formed by the arrangement of the hepatocytes. Each cord is having the rows of the hepatocytes, and these hepatocytes are hexagonal in shape. Get up. Stand up and leave the class immediately, both of you. Get up. Take your bags. I say get out immediately. I don't want to see such foolish students. Out. What's your roll number? 44. Good. Out. Now look this part. This is the one lobule. You are seeing this is the central vein. Radiating from the central vein, I will see your laugh. Okay? Go. This is the central vein. Radiating from the central vein, you are seeing the rows of cords. In each cord, the hepatocytes are present. So, when we see the lobule of the liver, you will find such picture. In the center is the central vein. Radiating from the central vein are the hepatic cords, and this is the periphery of the lobule. At the periphery of the lobule, you are find the triangular structure. These triangular structures are called the portal triad. So, in the center of the lobule there is present central vein, and radiating from the central vein are large number of the hepatic cords. Each hepatocytes, they are hexagonal in shape, and at the periphery of the lobule you are finding the triangular structure, that is called portal triad. Why we named this as portal triad? See this diagram, it is having the three structures. One is the hepatic duct or the bile duct, another is portal vein, and third is the hepatic artery. So, hepatic artery or hepatic arteriole, portal vein and bile duct, get band karo. These are the three structures present in the portal triad. When we see the slide of the liver, you will find all these structures. Then the examiner will ask you each point. What is this? What is this? You have to reply. Okay? That this is the portal triad. The structures present in the portal triad are these three, and these are the rows or cords of the hepatic cells or hepatocytes. Now I am coming to the another point. On one side of the hepatic cord there is the space. You are looking the space on each side of the hepatic cord. These spaces are containing the cells. These spaces are called hepatic sinus. These spaces which are present on each side of the hepatic cords are called the hepatic sinuses. And in the hepatic sinuses there is present the cell that is called Kupffer cells or Kupffer cells, what you like to pronounce, depends on you. Okay, so Kupffer cells, they are the cells which destroy the hormones or any the waste material of digestion of the food. That means Kupffer cells, they are showing the protective functions. They can destroy the another part which is not important for this. So that is the structure seen in the slide of the lever. Whole of the lever is signed by the capsule, that is Gleason capsule, and from the Gleason capsule the septa are coming and they divide the lever into the lobules, and this is the structure of one lobule. What is this? Portal tract. And look this part: hepatic artery, bile ductule, and portal vein. Again, the picture of the portal lobule. And look this part. This, how many lobules are here? One lobule, another lobule, like this. In each lobule, you are looking the central vein. Okay. Now, see this triangle. It is connecting the adjacent lobule, the central vein of one lobule, central vein of another lobule, central vein of another lobule. So, the adjacent lobule or the structure connecting the central vein of adjacent lobule, that is called portal lobule. Look diagram. This is the portal lobule. This central vein is connecting with the adjacent central vein, and this central vein connecting adjacent central vein. Now, see this portal lobule with the another hepatic lobule. Any difference? Diagram देख के बताइए. Is there any difference between central lobule and portal lobule? मौका मिलना चाहिए आपको. तो अब तो दिख रहा है. अब बताइए. Any difference? In hepatic lobule, there is center is the central vein, while in the portal lobule, in the center is portal tract. Okay. So portal lobule, in the center of the portal lobule is portal tract, while in the hepatic lobule, there is in the center is central vein. Because of that, I am showing you this diagram. Another point comes hepatic acinus. Look this one. You will read this in the physiology also. This is the part central vein, adjacent, and this is the portal tract. This is the portal tract, and it is connecting with this central vein, and two adjacent portal tract are connecting. This is the hepatic acinus. All right. Now look the another slide. Gallbladder. The function of gallbladder is storage of bile. Bile is coming from liver. The function of liver is two. Serous function, formation of the bile, and also it is also forming the plasma proteins. It is also forming the plasma proteins. So these are the two functions of the liver. The bile is formed by the liver. That bile is stored and concentrates in the gallbladder. So function of the gallbladder is storage and concentration of the bile, nothing else. Because of this, in gallbladder there is no muscularish mucosa, no submucosa, underline that. In gallbladder, no muscularish mucosa and no submucosa. No submucosa, no muscularish mucosa. Epithelium is lined by simple columnar epithelium, and fresh border is seen. So So large number of the mucosal folds you can see, and they are lined by simple columnar epithelium, and brush border may be seen. In the lamina propria, you can see the cut section of the villi and this. Look here, you are seeing the smooth muscle fiber, connective tissue, and blood vessels. That means fibrous tissue is there, muscular tissue is there. Muscle fibers are visible, and also the tissue, that is loose connective tissue and this. Because of this, this is called fibromuscular coat. So instead of muscularish mucosae, there is fibromuscular coat is present. All right. Suppose I am examiner. I will ask you why the muscularish mucosa is absent. What is the function of gallbladder? खड़े हो जवाब दो. क्यों? अभी मत बैठना फिर. बैठ जाओ. यादव. Sit down. जब तुम ही जवाब नहीं दे रही, ये लोग क्या बोले? Muscularish mucosa helps in the contraction of the villi. Muscularish mucosa is help in the contraction of the villi, and the part of submucosa muscularis externa, the contraction of the muscularis externa is help in the movement of peristaltic movement of the GIT. Peristaltic movement of the GIT is done by the contraction of muscularis externa, and contraction of the muscularis mucosa helps in the movement folds of the villi. Okay. The function of the gallbladder is storage, concentration. So no necessity of the muscularis mucosa. Clear? Contraction of the muscularis mucosa helps in the movement of the villi. यहाँ तो पर्पज ही दूसरा है. It is store. Function of the gallbladder is storage of the bile and concentrate. Because of that, no muscularis mucosa. So identification of the slide, you will not find. You will find just the folds, mucosal folds lined by the columnar epithelium, brush border. You may see no muscularis mucosa, and remaining part is same, serojoint, everything is same. That is gallbladder. Next is, look this slide, again very simple to identify. Pancreas. Again